If you refer to the definition of cell cycle, is the sequence of events that take place between one cell division to the next. Maksudnya, setiap sel dalam living things kan, mereka punya kerja utama ialah membahagi, undergo the cell division to produce the new cell. But this happening, they have the events. Dia ada susunan events tu, tak boleh berlaku sesuka hati. Will happen alternately. Dia berlaku secara bergilir-gilir untuk melengkap satu kita hidup sel. And the length of stages vary among species and type of cell. Vary maksudnya berbeza ya, different. So, waktu tempo mitotic phase dan tempo interphase adalah berbeza untuk setiap jenis sel atau setiap spesies. Contoh, okay, I always use the example, let's say I study the skin cell. Okay, the skin cell, okay, between the human, okay, with the animals from other classes, maybe such as reptiles. Okay, when we compare our skin cell, right, the life cycle show different length, okay, different length of stages, even though we study their covering tissue. Or if I study one species, let's say I focus on human only, but I look at their different cells, okay, skin cell or neuron or even muscle cell that we have studied in the chapter 2. Okay, I noticed that each type of cell have their own record or their own data of mitotic phase and interphase. I would like to start with the interphase of cell cycle because this is the longest stage in cell cycle. Dia ambil masa yang paling lama, hampir 90% of one cell cycle composed of interphase because they have 3 subphases. The first subphase is called as first gap phase. So this is the abbreviation, simbol dia yang mesti ditulis sedemikian, G huruf besar dengan 1 subscript. After the G1 phase, followed by the second phase which is the S phase. S huruf besar atau nama dia synthesis phase. Okay, in matriculation, okay, we call it as DNA synthesis because in this phase, DNA will be produced. Okay, another copy of DNA will be produced. And the last subphase of interphase is G2 phase, the second gap phase. Okay, second gap phase. So there are 3 subphases of interphase. G1 is characterized, the main event that can be observed significantly is the growth of cell. Okay, that means you will notice that after the first gap phase, right, the size of cell is larger. Okay, kenapa dia bertambah besar? Sebab the volume of cytoplasm increases. Isi padu cytoplasm bertambah dan ini memberikan fenomenon kita kata pertumbuhan sel berlaku. Okay, why the cytoplasm of the cell increases is because in the cytoplasm, there are a few organelles undergo duplication. Okay? They make many copies. They make many copies of themselves. This is why they contribute to the increment of the cytoplasm. Okay, why these organelles increase in the numbers? Kenapa dia bertambah dari segi bilangan dia? Sebab mereka nak fulfill, okay, the purpose that to produce protein, okay, which consists of enzymes. Okay, sebab we know that all enzymes are proteins, right? So they would like to produce the enzymes and some proteins for the process called as DNA replication. Okay, because DNA replication is the event happens in S phase later on. Okay, maksudnya dia membuat persiapan untuk sel tersebut melangkah ke S phase. Dan S phase adalah satu proses memerlukan banyak enzim dan protein di mana.
Ribosome?
Yeah, this is the ribosome. Okay, ribosome because they involve in the synthesis of protein. Yeah? So ribosome is the organelle that provide or they are actually produce the proteins. And most proteins will be converted to become enzyme involved in the DNA replication. Okay, so ribosome will be increased in number through process duplication.
Mitochondria.
okay, what is the main function of mitochondria? Why they are undergoing the duplication process?
Produce ATP.
Powerhouse.
Because ATP is the form of energy required by the cell to perform the following events. Okay. Another thing is we focus on the nucleus of the cell. Okay, the nucleus of the cell is already they have the chromosomes. Okay, they have the chromosome, but the chromosome is in chromatin form. Chromatin is the term describe the DNA in a thin structure, thin fiber structure. That's why kita kata dia loosely packed. It's not yet undergo condensation process. Dia belum mengalami proses yang kita kata coiling yang hebat tu. Dan sehingga dia boleh membentuk struktur yang dinamakan kromosom. Okay. So chromatin is the term to describe the DNA when they are still in the thin fiber form, okay, loosely packed. But after they undergo the coiling process, they will be called as chromosome because they able to be observed under microscope. Okay, now I move on to S phase. Yeah? Okay, S phase, S for synthesis. So what is synthesized here actually is the DNA. Okay, so again this is this is a nucleus ya. Yang ini bukan membrane of the cell ya. Ini adalah nucleus. So yang border ini adalah nuclear envelope. In the nucleus, that is the chromosome. Okay, and chromosome here. Okay, chromosome consists of DNA. Here is a pair of chromosome. They are same type. Okay, kenapa saya tahu dia same type? Sebab dia disimbolkan dengan huruf A. Maksudnya dia membawa gene A. Okay. Okay, so kromosom ini dia terdiri daripada satu, kalau lukiskan satu kromatik sahaja. Okay, di sini satu kromatik saja. Maksudnya dia membawa satu unit DNA. Okay, satu unit DNA. Okay, apa yang berlaku semasa S phase ialah, now each unit of DNA, they will undergo process replication ya. Again, this is produce copy. Dan kamu akan nampak at the end of the S phase, right? At first, the chromosome have one DNA. Now they developed another one. So this chromosome have two DNA. The pink color also, they carry the gene A, but recessive A ya, huruf kecil A kan. Okay, originally they have one DNA. Okay, now they undergo replication, they produce another DNA. Okay, so now we can say that the chromosome after the S phase, the chromosome is duplicated. Duplicated maksudnya kromosom sekarang kan, dia membawa dua sister chromatic. So this is two sister chromatic. Okay, ini pun sister chromatic. So macam mana saya tahu dia sister chromatic? Kalau DNA tu dia dipegang oleh same centromere, okay, they are sister chromatic. Kita punya sister chromatic, dia punya makna ialah kita punya dua-dua sister chromatic untuk satu kromosom itu yang telah mengalami DNA replication tu, original tengok dia ada satu DNA saja. Okay, dia mula-mula dia satu. Okay, iaitu yang ini. Okay, mula-mula dia satu DNA saja. Tetapi bila lepas proses DNA replication kan, they will develop the second DNA. Okay, the second DNA which I draw with the blue line. Okay, how I label the sister chromatic is when the identical sister chromatic, usually they are attached at the same centromere. Okay, so dua ini adalah dilabel sebagai sister chromatic sebab dia di centromere yang sama. Sama juga untuk dua ini, dia sister chromatic. Tetapi ini boleh ke? Tak boleh sebenarnya. Tak boleh ya. Sebab kenapa? Dua ini dia terikat kepada centromere yang berbeza. Okay, ini pun tak boleh jadi sister chromatic sebab they are the DNA atau chromatics yang we say attached at different centromere. So S phase, the main point is we need the student to understand actually what happened in the nucleus is the chromosome originally they only have one DNA. Setiap kromosom satu DNA sahaja mula-mula. Okay. Setiap kromosom satu DNA sahaja. Tetapi lepas S phase, satu kromosom dia ada dua DNA. So macam mana saya tahu dia dua DNA? Sebab saya kira dia punya chromatics itu. So DNA boleh dikira dengan berdasarkan chromatic dia. Okay, so sini ada dua. Setiap kromosom ada dua. Okay. Second gap phase. Okay, lepas kita punya S phase, right? Second gap phase, they continue the process like quite similar with the gap one. Tapi dia bukan growth phase ya. You can see they still focus on continue the process of protein synthesis and also the energy acquisition phase. Okay, dia akan menghasilkan banyak-banyak energi dalam bentuk ATP. Okay, tujuan dia ialah get ready untuk mitosis. Okay, the next event in mitotic phase. So G1 and G2 seems like they are preparing for the next event. Yeah? This is why we focus on why the ribosome need to be duplicated, and same goes to the mitochondria is highly duplicated. Okay. Kalau saya salah tulis di sini. Kalau organelles atau structure ya, kita gunakan istilah duplicated. Dia membuat salinan. Okay. Tetapi kalau dia molekul macam DNA, kalau dia buat salinan, kita guna istilah replication. Okay. So this is the usage of the terms. Even though both bring the same meaning, dua-duanya membawa maksud sama iaitu membuat salinan. Okay, tetapi untuk second gap phase ada satu organelles yang kita kena fokus, which is centrioles. And centrioles is only found in animal cell. Okay, bukan semua cells ada centrioles. Okay, so in the G2 phase, centrioles will undergo duplication also. So in one centrosome is already have a pair of centrioles. Okay, now each of it able to duplicate, right? So you will notice we have 4 centrioles now. Okay, but they will undergo a maturation process, iaitu they will separate. Okay, they will separate. These two will separate in one group. So they have their own centrosome. So now we have 2 centrosomes, and each centrosome have a pair of centrioles. Okay, just we always draw with this symbol. Centrioles always found in a pair, and they will arrange perpendicular to each other. Okay, dia akan susun sedemikian ya, sudut tegak. That's why bila kita buat cross section of the cell kan, satu bagi keratan memanjang, satu bagi keratan yang macam ni, merentas punya. Okay, so this is the significant events in the G2 phase. So overall, the interface of cell cycle has 3 surfaces, and because of these 3 surfaces, they contribute to the longest stage in the cell cycle. Okay, mitotic phase selalu gunakan simbol M huruf besar. Okay, mitotic phase ni terdiri daripada mitosis dan juga cytokinesis. Okay, so dua ni adalah proses yang berlaku dalam mitotic phase. Okay, this is a simple diagram to let you all to understand what is meant by life cycle of cell. So katakan I mula dengan satu sel ni ya, I start with this cell. After the first gap phase, I should use term gap ya. First gap phase, right? Okay, the main event in first gap phase is the cell growth. Okay, sel mengalami pertumbuhan. That's why gunakan satu tergambar menunjukkan pembesaran size. Okay, the cell become larger now. Okay, the cell will continue to the S phase, synthesis phase, where we have to focus on the activity in the nucleus. Each chromosome found in this nucleus, okay, before the S phase, they only carry 1 DNA. But after S phase, they carry 2 DNAs because they are become duplicated chromosome. Okay, chromosome yang kata ada 2 sister chromatics. Okay, this cell will continue to the second gap phase. Okay, second gap phase. Okay, the events quite similar with the first gap, which still focus on the reason why ribosomes are duplicated and also the mitochondria, because they are going to use both proteins and the energy in the mitotic phase. But kalau animal cell, they involve the duplication of centrioles. Okay, so maksudnya di sini dalam cytoplasm ada satu centriole sahaja, satu pair centriole sahaja. Sampai sini satu pair centrioles. Tapi after the G2, right, the cells have two centrosome dan setiap centrosome tu ada satu pasang centriole. Itu maksud dia. Okay, now barulah kita study cerita dia masuk ke mitosis dan cytokinesis, di mana after the mitotic phase, right, this cell will divide into two total cells. Divide into two total cells, and because they undergo division, right, so usually the total cells has smaller size. Okay, size dia lebih kecil. But each total cell able to start their new cell cycle. So setiap satu boleh mengalami new cell cycle. This is why I say is a sequence of events occurs between one cell division to the next.